I'm Amanda, and today I'm going to talk about an extremely important process that is happening in your body all the time, and this is DNA replication. So, first things first, DNA replication is semi-conservative. And what that means is when DNA replicates, one of the strands is from the original DNA molecule, and the other strand is a new strand, and that strand is called the daughter strand. So one of the strands is been preserved and the other one is a new strand. So what we're witnessing here is the original DNA molecule basically being unraveled and separated into two strands. And this is done by an enzyme known as helicase. What helicase does is it basically splits DNA in the middle by weakening the hydrogen bonds between the nitrogen bases. The part where the DNA molecule is being split into two is known as the replication fork. So what we're witnessing here is called the replication fork because it kind of looks like a fork. So replication runs in opposite directions. This top strand here is called the leading strand, and the bottom strand here is called the lagging strand. DNA replication in the leading strand is very simple. As explained in the DNA structure molecule, DNA is anti-parallel. So it runs, the two strands here run in opposite directions. In this leading strand, the parent strand runs from 3' to 5'. And so when it replicates from 5' to 3'. This makes DNA replication very simple because the enzyme DNA polymerase III always replicates from 5' to 3', and so the steps in which the leading strand replicates is extremely simple. Basically, DNA polymerase III assembles new nucleotides to the exposed DNA in the 5' to 3' direction, and it synthesizes continuously. So as you can see, this is DNA polymerase III, and it is synthesizing the new DNA on top of the old parent strand. And this strand here is known as the daughter strand. As you can see, the old parent strand here is being exposed, and it serves as a template for the new nucleotides to be assembled. As we said in the previous video, we know that adenine is always pairs with thymine, cytosine always pairs with guanine, and basically the new daughter strand, recognizing that these are the base pairs, are able to assemble knows what bases go on top of what, and are able to synthesize the new daughter strand on top of the old one. Now the lagging strand is a little more complicated in terms of replication. The lagging strand is synthesized discontinuously because, as I stated before, in the lagging strand, the parent strand is from 5' to 3', and thus when the new DNA molecules are synthesized, or the new daughter strand has to synthesize on top of this lagging strand, it has to synthesize from 3' to 5'. And since the enzyme DNA polymerase III only synthesizes from 5' to 3', this makes the lagging strand having to synthesize the new DNA in a series of steps. So in the first step of DNA replication in the lagging strand is an enzyme known as RNA primase synthesizes an RNA primer, and this basically serves as a starting point for the DNA polymerase III to later synthesize the new nucleotides. So as we saw before, if we go back, this enzyme here, this pink enzyme here, is RNA primase 1, and this primase basically synthesizes the RNA primer on top. After that, this molecule here, known as DNA polymerase 3, uses the RNA primer as like a starting point and synthesizes DNA from this primer to the next primer, and basically synthesizes from a 5' to 3' direction. So it's assembling nucleotides continuously until it reaches the next primer. So as you can see, these new nucleotides synthesized here are being synthesized between the two RNA primers, and this whole thing here is called an Okazaki fragment. So yeah, after that, DNA polymerase 1, so this is that green hand-looking thing, replaces the RNA primer molecules, nucleotides with DNA instead of RNA. So yeah, it's replacing the RNA with DNA nucleotides. It's replacing them, and again, just like in the leading strand, this parent strand here acts as a template for the new nucleotides to be assembled on top of. So this light blue strand here is also called the daughter strand. So it's replacing the RNA and putting DNA there instead. After that takes place, you see this little gap here. That's a no-no. So as a result, another enzyme known as DNA ligase comes and basically fills in those gaps with more nucleotides. And it basically assembles these Okazaki fragments together. And after that, you basically have your DNA is replicated, both in the leading strand and the lagging strand. Thanks for watching!